Good morning, everyone. Thanks for coming. My name is Lixin Wang. I'm the vice chair for the Asian Ecology section for ESA. This is ESA Asian Ecology section talk series. So the idea is we try to bring highly accomplished Asian scientists from the globe and try to have a talk series every 2 months to talk about different topics in ecology. So this time we are really happy to have a Professor Lingli Liu here to talk about her research. Dr. Liu is a professor and principal investigator at the Institute of Botany, Chinese Academy of Sciences. Her research focuses on the ecosystem carbon and natural cycles and their response to global environmental changes. Dr. Liu has been published widely, and she was awarded the National Natural Science Foundation for Outstanding Young Scholar in 2015, and then she received even more higher distinction, the Distinguished Young Scholars Award in 2021. She is a chief editor for the Chinese Journal of Plant Ecology. She also served on the editorial board for several international journals, including Ecology Letters, Global Change Biology, Journal of Plant Ecology, and Fundamental Research. So the talk will be recorded, and we typically put the talk on the ESA website. So in case people can't make it, want to revisit the talk again, you can go to ESA website to look for this talk. Dr. Liu, I forgot to ask you, is that okay?
Sure, it's okay.
Yeah. All right. So yeah, without further ado, I'll give the podium to Dr. Liu. Thank you again for being here. We are really happy to have you here.
Okay, thank you, Lixin, for the invitation, and good evening for everyone. I say most of us joined online from the United States. It's been a great pleasure for me to give this talk. And well, first I want to express my thanks for Lixin and for Jiafu and for all this AES and board member for the invitation. And I actually, I think I have been AES member maybe more than 15 years, even longer, should be longer, because I have this served as a secretary when Professor Hu was a director, and also I say Professor Tian is also online, and also worked with them during when they chair this Asian Ecology session. So it's been so wonderful to reconnect with everyone. Even it's online, and I see a lot of familiar faces and friends. And also, I missed the chance to talk with everyone for the several ESA meeting, but it's so great to see so many people come online. Anyway, now let me introduce what I want to talk today. So today I'd like to share our research progress about how nutrient strategy of different mycorrhizal plant. We know mycorrhizal association is very fundamental for plant nutrient acquisition, could have significant impact on soil carbon storage. So in this talk, I will particularly focus on how different nutrient acquisition strategy of EcM and EcM plant could influence their carbon nutrient acquisition. Seems my slide. Oh, okay. Now I'd like to give an overview of what I will cover today. And so first, I wanted to discuss how evolution and ecological force could shape the nutrient acquisition strategy of different mycorrhizal plants. And in the second part, I'd like to share the insight from our recent synthesis to show how interconnect mycorrhizal could enhance the interplant nitrogen sharing. The third part of this talk will about experiment we conduct at the BF China. Here we wanted to show whether the different mycorrhizal symbiosis could influence the plant productivity in high diversity ecosystem. Finally, drawing from a study we conducted across from North to South China, I want to explore how different mycorrhizal acquisition strategy could influence soil carbon storage. So why mycorrhiza have been so important? We know mycorrhiza have been a result of co-evolution between plants and fungi. This evolution is occurred from 450 million years ago. The early terrestrial plant have been formed the symbiotic relationship with EcM fungi. And after that, about 180 million years ago, plants will be also evolved another mycorrhizal type is EcM fungi. Those mycorrhizals particularly crucial for plant to acquire carbon and acquire nutrient and also waters. And we know about 85% of terrestrial vascular plant have been formed mutualistic symbiosis with mycorrhizal fungi. So the partnership will be particularly important for the EcM and also EcM. The two mycorrhizal fungi have a very different nutrient acquisition strategy. I will explain the detail in the later slides. So this different strategy have been mentioned, gave a good summary by Phillips, new phytologist, technical review paper. So you can see for the differences, particularly different is the EcM fungi have limited subtrophic ability. So this means they have to obligate to symbiosis with plants and depend their host to deliver the carbon to the soil process, and then this make a partnership with subtrophic microbials. This promote subtrophic microbial decomposition, released inorganic nitrogen, and then EcM fungi can help the host plant to acquire those inorganic nutrients. So for EcM plant, this inorganic nutrient strategy also generally have those host plant have a higher litter quality. But differently for EcM fungi, EcM fungi have the ability to produce enzyme. This means EcM fungi can acquire organic nutrient from the soil, and those acquired organic nutrient actually result in competition with subtrophic microbes. So the organic nutrient of EcM fungi will also accompany with plants have lower litter qualities. The different nutrient acquisition strategy maybe can explain why EcM and EcM fungi have not only different EcM and EcM plant not only have very different trait, but also they have different distribution. The Nature paper published in 2019 shows EcM and EcM distributed are very different in across the different biomass. So for EcM plant, it particularly dominate in the tropical and subtropical region. Those EcM, this helping EcM have in organic nutrient strategy have crucial for them to uptake soil from the nutrient poor tropical subtropical soils. But for EcM plant, their primary distributor can be found in temperate boreal forest. Here, the EcM, the organic nutrient strategy, will be facilitate those plants to uptake nitrogen from the nutrient-rich organic source. So this different distribution between EcM and EcM potentially suggests the two mycorrhizal types have a very different strategy, and this could influence ecosystem function and also their adaptation. So with all those information have been mentioned, we want to ask the first question is how evolution and ecological force have been shaping the nutrient strategy of woody plant mycorrhizal symbiosis across different biomass. To address this question, my PhD student Lulu Guo have conducted a global synthesis study. In this study, Lulu acquired about 500 woody plants, and also she collected data including leaf and root trait, and also their decomposition rate and the resorption rate. So what her result found is compared to EcM plants, AM plants showing more acquisitive root and leaf trait. This means for AM plants, it have higher specific leaf area, higher nitrogen, lower lignin concentration, and for roots, it have lower root tissue density but high nitrogen and high phosphorus concentration. So this means not only for the leaf but also for the root, AM plant shows more acquisitive trait. And when we look at the different the change across the evolution time, just with the increase of diverging time, this here shows some older species and this recent evolved species. If we can see the different for the blue one indicate the AM species and the red indicate the EcM species. If we look at the difference, we can see AM species consistently maintain the higher nitrogen concentration, but for EcM species, even the older EcM species have lower nitrogen concentration, liter concentration. This means more conserved trait. With over evolution time, we can say EcM becomes more acquisitive. This means its increased nitrogen concentration have been increased. Similarly for specific root length. So for EcM is involved in the older EcM species. It actually have thicker roots, and but recent developed EcM species have the roots become thinner and longer. So this means with the evolution time, even EcM still have lower and more conserved trait, but it can become more acquisitive. So how those shift in leaf and root trait could result in the different nutrient acquisition strategy. First, I'd like to talk about how plants get their nitrogens. Generally, plants get their nitrogen through two pathways. The first pathway is the resorption process. The resorption process happened during fall. When during fall season, leaf have been resorbed the nitrogen in their tissues and go back to the triggers to storage for the next years. And when early spring come and the leaf use those nitrogen stored in this tissue to supply the early development of the leaf during springtime. And then another part also generally the major part is come from the root uptake, but it's only account of 50 to 60% across all species. So the two pathways together determine how much plants can get their nitrogen. So when we look at the two different pathways, how the evolution changed for the AM and the EcM species, and the result is consistently interesting, is consistent with the EcM plants become more acquisitive, and actually their nutrient acquisition pathway is become more acquisitive. This is because compared to the older species, younger, the recently evolved EcM species, it's have a lower nitrogen resorption rate. This means it's become less dependent on the resorption. This more conserved pathway, but its decomposition rate have been increased. Then this means EcM plant become more dependent on the mineralization process. So the results indicate over time EcM species also not only its trait become more acquisitive, and it's also shifted toward to a more nutrient acquisition strategy. This is depend on decrease resorption, but increase the nitrogen supply by the decomposition and mineralization process. Then could this different nutrient strategy explain their distribution between difference between AM and EcM plants? For those nature papers, it shows decomposition is the most important pathway. And indeed, consistent with this prediction, with the increased decomposition, this means more inorganic nutrient can be released in the soil. And for this inorganic strategy of EM, it's been more suitable for their nutrient acquisition. But also for the EM species, if we say for the tropical region, we can say EM species actually also not only have higher decomposition rates, but it's also have higher resorption rate, the blue one. So this suggests over time, this suggests in warmer and more humid region, EM species not only have more nitrogen released during decomposition, but also can acquire more nitrogen by the internal process. This means the resorption process. The two strategies together suggest the EM species maybe more developed more effective nutrient acquisition strategy in the tropical region, and this will partially explain why EM species generally distribute more in the tropical regions. So as mentioned earlier in this study, this mycorrhizal association probably not only have been more benefit a lot or for the individual species, but also for those mycorrhizal, we know it can actually form the interconnect mycorrhizal network. So this means the nitrogen symbiosis, plant symbiosis with the nitrogen with mycorrhizal probably not only benefit the plant itself, but also maybe they can share with their nutrient and resources through the interconnected mycorrhizal network, and then it can also benefit with their neighbor plants. So in this second part, we'll ask a question: could this interconnect mycorrhizal network have enhanced interplant nitrogen sharing? But in particular, this mycorrhizal network sharing have been gained a lot of attention in the past 2 to 3 years. Here I'd like to mention the Tedros 2020 paper. In this paper, review paper, I particularly mentioned when mycorrhizal plants form the mycorrhizal network, and those trees tend to share their resource also not only nitrogen, but also the cycle between such as drought stress, also insect attacks. The signal can be shared via mycorrhizal network to their kin species, or at least for the same type of species. But this common mycorrhizal networks hypothesis have been received a lot of debate. Like in the Nature Ecology Evolution paper, it shows that maybe this common mycorrhizal network concept is just because of the positive citation bias. Anyway, a lot also the NUFA paper and function ecology paper also discussed why how the mycorrhizal network whether it exists and why what the processes control this nitrogen sharing between plants. But actually, this mycorrhizal network could promote plant nutrient sharing have been studied for many years, but even have been conducted a lot of study. There is a lot of quantitative synthesis to put those information together. So my PhD student Yuxuan Gao have conducted a synthesis together to put all those nitrogen interplant nitrogen sharing study together. This data structure is kind of quite complex, so I won't go details about this, but we can say in general those studies include for nitrogen labeling for donor plants, and by manipulate the mycorrhizal network and to see if with intact mycorrhizal network how much nitrogen can be received by receiver plants, or if we disturb the mycorrhizal network, will this be reduced? Using this data set, Yusheng want to test several case hypotheses. Particularly, this hypothesis, I want to explain two key hypotheses we're trying to test. So the first hypothesis is come from the mother tree hypothesis. The mother tree hypothesis indicate for those species with close phylogenetic distance, then more nitrogen have been transferred. So this means the larger or older tree tend to transfer more nitrogen to the seedling, their young seedling, or at least their close kin. Then with the increase of phylogenetic distance, nitrogen transfer should be decreased. Another hypothesis we want to test in this synthesis is source-sink relationship. This means if the donor and the receiver plants, they have higher nitrogen grading, and then the source sink, the larger source sink grading, means more nitrogen can be transferred from donor to receiver plants. So what did she find? Indeed, Yuxuan's data set showed symbiosis with mycorrhizal fungi and nitrogen fixed symbiotic can enhance the interplant nitrogen sharing. So this data shows for the green ones shows for plant without any symbiotic with soil microbes. For the green ones with the intact mycorrhizal network, we can see the amount of nitrogen transfer from donor to receiver have been increased about 72-fold. Without mycorrhizals owning nitrogen fixation bacteria, this nitrogen sharing also increased by only 2.6-fold. But interesting, when those plants symbiotic with mycorrhizal network and also nitrogen fixation bacteria, the amount of nitrogen have been increased about 9.4-fold. So the nitrogen, the result also shows the nitrogen sharing is strongly influenced by source sink grading. So when nitrogen the nitrogen grading between the donor and the receiver plant increase, this is the log ratio. If this grading become more stable and then symbiotic with mycorrhizal network, also mycorrhizal and nitrogen fixation bacteria together will result in significantly higher nitrogen sharing from receiver plant to from the donor plant to the receiver plant. But interestingly, we did not find the phylogenetic distance will affect the amount of nitrogen sharing between the donor and the receiver plant. If we only have mycorrhizal network or only have a nitrogen fixation bacteria. But if the two symbiotic relationship combined together, the result shows with the increased phylogenetic distance, then more nitrogen can be transferred from the receiver plant to the donor plant. From the donor plants to the receiver plants. This result is actually different from the mother tree hypothesis because we use with the mother tree hypothesis, suppose this interplant nitrogen sharing should be declined. We observed an opposite trend, and in further data analysis shows this increased trend actually more driven by the nitrogen grading difference between donor and receiver plants, because for the nitrogen fixation combined with the nitrogen fixation, receiver plants generally have higher nitrogen concentrations. So the result together shows indeed mycorrhizal collection will promote interplant nitrogen sharing, but those nitrogen sharing particularly will be driven by the source sink relationship. So this suggests the mycorrhizal network could promote plant coexist and also probably improve ecosystem biodiversity and functions. However, in this study, most of those experiments have been conducted in the labs, in laboratory, and particularly in greenhouse. But in the nature ecosystem, it's become more complicated. So whether the plant with different mycorrhizal network could affect the ecosystem function, particularly for the biodiversity, its different mycorrhizal association will influence different for the plant competitions. So to address this question, we conduct another field experiment at one of the largest biodiversity and ecosystem function experiment in China. This is called BF China. It is a subtropical forest site. In this BF China's experiment, we wanted to test how different nutrient acquisition strategy between EM and ECM plant will influence the competition in the high diversity ecosystem. In January, we know with increased tree diversity and plant productivity will increase. This BF positive biodiversity ecosystem productivity relationship have been proved in BF China site. This is Huang Yu's science paper in 2018. So it shows with increased tree diversity, standard volume increased. And this has been true not only site A, but also site B. And particularly it's interesting since 2013 to 2017, we can see the slope being increased more steeply. This means the positive relationship between biodiversity and productivity becomes strengthened with the increase of years. So for the ecosystem, if we wanted to, it needed to maintain the positive relationship between biodiversity and productivity. This means the plant have to have enough nutrient supply to maintain this positive relationship. And then to maintain the positive relationship with those nutrients either come from resorption or from mineralization, like we mentioned before. So in this study, we wanted to test with increased biodiversity, how plant community will change the two pathway, and will AM and ECM species may be different. And to do so, we conduct we measure little resorption and also decomposition at the BF site. And I like briefly introduce about the experiment design for BF China. In BF China, there are 5 biodiversity levels: 1, 2, 4, 8, and 16. And in each biodiversity level, although this experiment not initially not designed to test the difference between AM and ECM species, but in each biodiversity level, there are just split half are AM species and half are ECM species, such as for biodiversity level at 4, there's 2 AM and 2 ECM. For 8, there are 4 AM and 4 ECM. And for 16 is also half 8 are AM and half are ECMs. So this experiment setting will give us a wonderful opportunity to test whether the different nutrient acquisition strategy play a way to regulate the biodiversity and the ecosystem response, ecosystem function relationship. And what we found is our results showed with the increase of tree richness and the NPP of the whole community being increased. This finding is consistent with Yuan Huang's science papers. But when we split this ecosystem at 2 sub-community, including the AM subcommunity and the ECM subcommunity, the 2 species subcommunity shows a quite different response. For AM subcommunity, we found with increase of the tree diversity and the NPP have been increased significantly. It's consistent with the whole community response. But for ECM, we can say with increased tree diversity, ECM subcommunity productivity declined significantly. And particularly we see at a species diversity level at 16, those ECM tree species here are being declined up, only account 5 to 10% of the abundance. This means after 10 years, even initially those 2 there are 8 species of the 16 community have been for ECM. It initially is half between AM and ECM, but after 10 years, those ECM have been out-competed from the whole community. So what makes the different response between different response of AM and ECM? Like we mentioned before, we wanted to see whether this difference come from their different nutrient acquisition strategy. So we split this acquisition strategy for the 2 pathways. So for the first resorption part, we tested the NRE is nitrogen resorption, community level nitrogen resorption rate, and PRE is the phosphorus resorption rate. And we also test the decomposition part. So the result shows with increased tree diversity, NRE and PRE have been increased significantly. And also for the decomposition is also increased significantly. So at a community level, this means the community also indeed have a more efficient pathway to acquire nutrients, not only by resorption process, but also by decomposition process. And then we can see that AM, but the ECM community will respond differently. For AM community, it's consistent for the whole ecosystem. The three pathways have been increased: resorption, higher nitrogen phosphorus resorption, and faster decomposition. But for ECM, the response is different. The ECM, the resorption rate have been declined significantly, both the NRE and the PRE, and the decomposition have been increased. Here I want to emphasize for plant, for ECM, if they increase the decomposition rate, this means more nitrogen have been released to the soil, and those nitrogen released during the decomposition process can not only uptaken by ECM plant, but also by AM plant. So this means ECM are being released more nitrogen back to the whole community, but it's returned less by themselves through the resorption process. And compared to AM, this means ECM species have developed less efficient nutrient acquisition strategy with increased tree diversity. Then we further to do the microbial community composition analysis by measure the fungi diversity. And those CCI analysis is quite complicated. I won't go details, but using those information, we also compare whether soil microbial community change have been partially explained the different response between AM and ECM. So for this structure equation model shows microbial community composition change have no significant impact for the overall response. But for ECM community, the trace species richness induced microbial community change have been partially explained the elective response of ECM trees. This is particularly true because ECM, we know ECM species have heavily relied on their fungi community to acquire nutrient strategy. So the result shows overall for the BF channel shows for the different response between AM and ECM species actually come from their different nutrient acquisition strategy, and also particularly for ECM microbial community composition change can significantly driven their different response. For those findings from BF channels focus on the above ground process, and we know a lot of carbon in the ecosystem actually stored in the soil. So this brings our to the next question is how different mycorrhizal strategy can influence affect the below ground process, particularly soil carbon storage. So here to set the stage, I would like to use a phrase annual review paper to develop this hypothesis. For AM species, we know it did not have the subtrophic capacity. So to get the nitrogen from the soil, there's a mycorrhiza needed to release the carbon supply, particularly from photosynthetic carbon to the subtrophic microbes. Then those subtrophic microbes use those carbon to degrade those organic matters, and then after release the nutrient, and plant can uptake this inorganic nutrient. This process will lead to soil carbon decomposition, is called the priming effect. But for ECM, this process has been quite different. We know ECM have maintained can release the enzyme, so it can acquire nitrogen directly from the organic matter. So this, this organic nitrogen acquisition strategy makes those EcM fungi actually out-compete with the subtrophic microbes, because this subtrophies also need nutrient from the decomposition from the organic matters. If the EcM fungi have been directly uptake those nutrients to the host plant, then it will reduce the subtrophic microbes activity, and then this will reduce the decomposition rate. So this process is called the Gauge effect. If we compare the two different strategies, this probably means M and EcM fungi have different nutrient acquisition strategy, and it can have different impact on the organic carbon decomposition process. But to quantify the contribution of mycorrhizal fungi to how much they acquire nitrogen to host the plant can be quite challenging. So in this study, we use a possible way to do it is to use the N15 transfer discrimination process during nitrogen fungi acquire nitrogens. This because mycorrhizal fungi tend to deliver nitrogen depleted, N15 depleted nitrogen to plant, and it returns heavily nitrogen to the fungi biomass. So we use the difference between this difference between plant and also fungi N15, and then we can estimate how much of nitrogen plants rely on through the mycorrhizal acquisitions. And to test how different mycorrhizal strategy influence soil carbon storage, we conduct an experiment from Changbai Mountain to Dongling Mountain, Dinghu Mountain to Xishuangbanla. It's from North China to South China, and there are also come with a shift from AM species to EcM species. So in this study, we for the 4 site, we have 3 sites have canopy crane, so we can access top canopy trees using sample their leaves, and we also can sample those trees below ground near the trunk about 1 meter away to get the soil and liters. And then we use the delta N15, is the difference between leaf and soil, to indicate how much plant rely on nitrogen from acquisition from mycorrhizals. So what we found here with the increase of, if the delta N15 become more depleted, this means we have more nitrogen derived from mycorrhizal acquisition. So the result shows with the increase of EcM fungi, indeed the plant have been more dependent on the organic strategy, and then more dependent on mycorrhizal to acquire delta N15, acquire nitrogens. And we also shows with the increase of EcM abundance, the soil nitrogen enzyme activity, the Vmax of the enzyme activity also increased. This, like we mentioned before, EcM fungi maintained the enzyme production capacity. So higher EcM abundance come with a higher nitrogen enzyme released. And we also found with increased EcM abundance and soil metabolic content have been decreased. The metabolic content is calculated by the microbial respiration divided by microbial biomass carbon. So with a high MMCO means there are lower carbon use efficiency for soil microbes, and soil microbes can produce less residuals. And so this result shows with the increased EcM abundance and then some MMCO decrease, and those communities will have higher carbon use efficiency. And we also found with the increased EcM relative abundance compared to subtrophies, the liter decomposition rate actually declined. Those means it's a stronger Gauge effect. So put this together, it means for EcM, for community with higher EcM abundance, it have soil microbial with higher carbon use efficiency and also a slower decomposition rate. And then put this together for those community with higher EcM abundance, we can say there have higher, not only for higher POM, but also higher mineral associated organic matters. So this means with increased EcM abundance, then the soil can storage the more carbons. And we also use measured those fungi community composition use high frequencies measurement, and those shows, and we try to explore what kind of ECM fungi explain this difference. And our result shows for those higher MAM and pure M community, this with ECM abundances, particularly with the medium distance explore type of hi-fi. So if we can say this for the ECM, for those, this means for the here this means for the more active responses, means plant more rely on ECM fungi to get nutrients. Then we can this particularly driven by those medium distances fungi. So and put it together, our result shows, the first result shows for the global scales strategy, the global nutrient acquisition strategy for AM and ECM fungi is not only affected by the its trait, but also the difference between the phylogenes and the climate. And the second part of it shows increased mycorrhizal fungi actually facilitate nutrient sharing among the plants. And the third part shows with different mycorrhizal nutrient acquisition strategy, both nutrient uptake by mineralization and resorption have been increased, and this being contributed to the competitive edge between AM trees over ECM in high diversity subtropical forest. And the fourth part, of course, the grading study, which shows ECM organic nutrient strategy actually promote the soil organic carbon accumulations. And now, finally, I'd like to thank my collaborator. I see Shui-Jing and Jiang, and Ling, Professor Jiang Ling is also online. Oh, actually mispronounced your name. Apologies. And also, I want to thank Professor Kaiping Ma for the BF China experiment, and also all my collaborators. And now I probably, now my talk is ending. So if you have any question, I'm happy to take it.
All right. Yeah, thank you, Professor Liu, for the interesting talk. Any questions from the audience? Please feel free to unmute yourself if you have questions, if you can talk, if you can using the mic, or you can type in the chat, and then I'm happy to read out too. So maybe I will start a quick question. So Lingli, for the second part, when you talk about the donor and the receiver difference, when they have a bigger difference, there are more nitrogen transfer, right? So that's interesting. I just curious how that related to the overall nitrogen level, right? So we can imagine, the donor and the receiver difference is one thing, but also the overall, maybe here is a low nitrogen, there is a higher nitrogen. So that maybe influence the transfer as well, right? Do you guys control that variable as well when you're doing the meta-analysis?
Could you repeat again? Sorry.
So I just thinking about, like the donor and the receiver difference, that is I can imagine, that is a big factor, is important. But also the environment, like they stay, right? So whether they stay in a higher nitrogen condition, or maybe they live in a lower nitrogen condition, right? So I guess even the difference is big, but if they live in high nitrogen condition, they just have a lot of nitrogen. I guess they just their relationship is different from when they live in a poor nitrogen condition. So I just in the meta-analysis, I can imagine the soil nitrogen level probably different. I just wondered whether that potentially play a role in your results, or you don't really differentiate them, you just put them together.
So you mean instead here you mean for soil condition if the donor plant is growing the high nutrient concentration will more nitrogen transfer? I guess so. In that but in those experiments most of those experiments are conducted in lab in greenhouse experiment. They use same soil. Similar, they use they actually. If I'm right, but I say you shouldn't collect soil nutrient as a random effect. But most when they start the experiment, they use the same soil, mixed the same soil and the plant and donor and receiver plant together. But I add N15 here.
Right.
Those are trace amount won't increase the total nitrogen supply. But as you mentioned, because there's a source sink grading, is if those part have a higher nitrogen concentration, and then the concentration grading will drive more nitrogen sharing to the mycorrhizal by the mycorrhizal network. Yes, if there are nitrogen concentration grading, I believe there will be more nitrogen and transfer from those higher section part to lower, just like water flow from high place to the low ground.
Right.
But it's a very good question. I think your hypothesis is correct, but those data, because to reduce those, you're more driven by the concentration between donor and receiver plant itself, themselves, instead of a soil concentration. Those experiments actually control this difference.
Right. Yeah, I mean, it makes sense. I just think about because the figure is scattered. I try to think whether you can find the different variable to control. I mean, to explain the scattered figure. I mean, yeah.
Yes, that's a good question, and I believe so. I believe so. That's why for those some experiment actually, when actually we had conducted another experiment using mycorrhizal, we put nutrient with a high nutrient little quality in a soil core, and we found the mycorrhizal actually can the mycorrhizal nutrient forge. And I know if many of you maybe know Evolutionary has the experiment actually can show the mycorrhizal actually can precisely find those nutrient rich part. So this means if soil have higher nutrient concentration, should have more nutrient transferred by the mycorrhizal network.
Okay. Yeah. Thank you.
Thank you.
Hi, Lingli. I have one question here.
Thank you, Professor Hui. Nice to see you.
Very, very interesting talk. Yeah, I have one question related to the productivity and the diversity. So yeah, so you share the results, in their study. So the AMPP increase with the diversity overall, and also when you separate into the AM and the ECM plants. Yeah, here this one. Yeah. So the AM plants showed the increase, but the ECM plants decreased. So here, so in your study, so you said different diversity, but you include both the AM plants and the ECM plants, right? Yeah.
Yes.
Yeah. So for the ECM plant response, what happens if you just grow them separately, just the ECM plant? If you set the different diversity, how the AMPP would respond? Yeah, that's my question here.
That's a very good question, and it's interesting for those experiments. We don't have a chance to test this hypothesis because for the diversity level here, they mixed half and half. But I think for AM probably the result. We know for the tropical region and the tropical region, and biodiversity promote ecosystem productivity. So for AM species, the higher diversity should promote productivity. But for ECM community, maybe the result is mixed, and we actually should very interesting to test the hypothesis you mentioned, because for ECM communities actually tend to form the monocultures communities, such as we know for this pine tree and for those typical ECM trees like the pine trees, and also for those oak trees that can still have some diversity, but particularly for pine tree, we tend to form a monoculture species, monoculture stand. And so for those monoculture standards come with this hypothesis, like the tidal source hypothesis for ECM community, because this tend to heavily depend on the host plant. Then those host plant interlinked with each other by the mycorrhizal network, and they share the resource, share the nutrient together. But those formation, if come from different species or different mycorrhizal type, and then the efficiency of mycorrhizal network for those community actually can decline. So probably for ECM species, increased diversity maybe have limited impact on productivity, or even have negative impact, but I'm not quite sure. I see Professor Hu is online. Is he online? Oh, he's quick. He probably can better answer this question than me, that I did. But that's quite a good question, quite an interesting hypothesis. And I didn't say. I know in Germany they have a mycorrhizal mycorrhizal BF experiment. In their experiment, the result actually, when we publish our paper, they also at the same time they also published a similar paper on ecology. In their result, the kind of opposite. In Germany side, in ECM community actually have higher productivity. But AM have been declined. They also mix this two mycorrhizal type together. But that's a part, the opposite response in Germany, probably because in Germany side is more temperate and boreal forest. So in that region, this means ECM, organic nutrient strategy of ECM become more promoted production. But I think your question is quite interesting, and yeah, maybe we can test this with some nature forest site data.
Okay, thank you.
Any other questions? Yeah, Renwei did have a comment that, yeah, Lingli, thank you for the nice work. Look forward to seeing more of your group's product in the future. So yeah, Wei is also a committee member for the AES.
So thanks everyone for attending this talk, and yeah, it's great to see everyone.
Yeah, it's great to see you. I guess, yeah, maybe one quick one. So what you talk about the resorption like a 30%, the mineralization about 60%. What about another 10%? Where?
Yeah, that's a good question. So that's why we say nitrogen for most ecosystem, nitrogen and most ecosystem productivity limited by nitrogen supply. So for in general, nitrogen unlimited, not 100. They need 100% nitrogen supplied, but the two major pathways cannot supply it. So such as nitrogen fixation, nitrogen deposition can supply the additional nitrogen those plant growth are related.
So the 10% either through natural fixation or through mycorrhizal fungi, you see? Is that
mycorrhizal fungi is most mycorrhizal fungi is actually account for the mineralization part or the root uptake part. But I mean for the outer source like nitrogen deposition and nitrogen fixation.
Okay.
It's not internal, not through the internal circle, but from outer side input.
Okay. Yeah, interesting. Yeah, I guess the idea is it's not 100%. They always suffer is less than 100%. Is that
yes?
Because the plants always has a natural limitation.
Yeah, they always have a nitrogen limitation, and so I stopped my screen here. So it can possibly lead to additional input, particularly for this why for those ecosystem has a high nitrogen limitation, and then they can mostly promote nitrogen fixation or nitrogen deposition why to stimulate plant growth because generally all ecosystems are nitrogen limited.
I guess, yeah, I know it's very early for you. Really, it's it's only 8:30 right now. You start at 7:30 a.m., I assume. So yeah.
It's 10:30.
Oh, I forgot. Yeah, that's the way around, right?
It's
Okay.
10:30.
Okay, yeah, right. Any other questions before? Oh, we do. I think, yeah, this from Professor Lin from Taiwan Normal University, right? Thank you for the impressive talk. It's very clear to me that your study has major implication on global change ecology. What would you say about how climate change may affect ECM, AM at a global scale? So I don't know, yeah, whether I can say it is yeah, basically question talking about what's your thought about the climate change impact on the ECM and the AM association in the future.
Yeah, this have been, I think there's also a hot topic being received a lot of attention those years, and we see a lot of paper published on nature or nature climate change geoscience. So in general, such as for elevated carbon dioxide, those research with increased elevated carbon dioxide, those studies shows probably ECM plant because it's a more organic acquisition strategy. And so in the nutrient limited ecosystem, like increase the elevated carbon dioxide concentration, then ECM plant maybe become more take advantage of increased elevated carbon dioxide. But for increased nitrogen deposition, then those input of inorganic nutrient to the ecosystem, this will be more shifted to the more favorable for AM species because those species have been more efficient to take inorganic nutrient, but the ECM can outcompete the ECM. And also, so in general, the global change can either increase nutrient limitation, like elevated carbon dioxide concentration in the favored ECM species, or it can increase nutrient supply, like increase nitrogen deposition, or sometimes for warming promote mineralization process, and then more favorite AM species. And also several studies shows land use change. Land use change tend to cut the ECM ecosystem to cause more disturbance for ECM ecosystem, such as convert the forest to to agricultural land, and those also result a lot lost of a lot of ECM abundance. Yes, definitely global change cause significant impact of the two different mycorrhizal type, and their response can be different depending on what kind of global change factor we're trying to test.
Yeah. Yes, thank you for the comprehensive answer. So any other questions before we finish? I didn't see any question now. Yeah, I guess, yeah, thank you again, Professor Liu, for the interesting talk. Yeah, that's
Thank
great.
you. Thank you, everyone. Oh,
Yeah.
good night.
And, yeah, thank you all for being here. Yeah, I will.
I'm here.
You guys, yeah, we'll see you
Okay.
all next time.
Bye bye. See a
Bye.
lot.
Thank you.